Good morning, everyone. My name is Dushani Palliyaguru, and I'm a postdoctoral fellow at the National Institute on Aging. Today, I will present to you some preliminary data examining the effects of riboflavin deficiency on Alzheimer's disease in the 5XFAD mouse model. Riboflavin, or vitamin B2, is an essential nutrient that is found in common food types like dairy, eggs, leafy greens, and nuts. Riboflavin is the precursor for flavin adenine dinucleotide, or FAD, which is an essential cofactor for proteins involved in a variety of different cellular processes. An ADPH quinone oxidoreductase, or NQO1, is one such flavor protein that is implicated in xenobiotic metabolism and other cellular homeostatic pathways. In vitro studies have shown that riboflavin plays an essential role not only in NQO1 stability, but also that mutant NQO1 isoforms co-aggregate with amyloid beta in cell-free experiments. The exact relationship between riboflavin, NQO1, and amyloid beta-driven diseases, including Alzheimer's disease, has not been examined in an in vivo model to date. In order to explore this further, we administered either control diet or riboflavin deficient diet to both wild type and 5XFAD mice of both sexes for 3 months since weaning. During the experiment, weekly body weight and food consumption measurements were collected, and at the end of the experiment, several metabolic and phenotypic measurements were collected prior to euthanasia for serum and brain sample collection. Moving on to the results, showed that riboflavin deficiency lead to reduced weight gain in 5XFAD mice. This was seen despite similar food consumption patterns. Also, 5XFAD mice appear to have stunted body weight compared to wild-type mice, an observation that has been reported previously by others. Riboflavin-deficient 5XFAD mice also showed reduced RER, a marker of energy metabolism and metabolic rate. Riboflavin deficiency is often associated with vision impairment in human populations, and therefore we measured this in our mouse cohort. For this, we utilized the vision portion of the mouse frailty index that examines features like corneal opacity, vision loss, Menis reflex, microphthalmia, and cataracts. Our results showed that riboflavin deficiency worsened vision impairment, and that was already present in the 5XFAD mice. Next, we measured levels of amyloid beta in the cortex of these mice using VPlex plus A-beta peptide panel 1 kit from Mesoscale Discovery. Riboflavin deficiency increased amyloid beta 42 levels in the soluble fraction of the cortex in both male and female 5XFAD mice. We have just begun also examining the specific role of NQO1 within the context of 5XFAD. And for this, we're characterizing 5XFAD mice that are completely depleted of NQO1. Preliminary data from these mice show that in their serum, so if you look at this, if you look at their serum, there tends to be an increased level of amyloid beta in the 5XFAD NQO knockout mouse compared to the controls. As a follow-up, we will be measuring amyloid beta levels in the brains of these mice, and also we plan on completing a full phenotypic characterization of these mice as well. With that, I'd like to thank everyone involved in this project, and especially thank the Intramural Research Program for funding this work. Thank you.